This session aims to go back to the basic principles of why we have standards and regulations and why we need to comply. To begin with the big why. What is the outcome that we are all working towards? The answer, I think, is that we are working to develop medicines and get these to the patients that need them in an efficient, cost-effective manner that sustains and grows our business. The first prime consideration in doing this is that all subjects and patients involved in clinical research should not be exposed to undue risks, and that their safety is our paramount concern. The link with subject safety is easier to see in clinics where we are directly administering drugs to humans and then caring for them during the study period. Labs also have their role to play, as lab results can reveal issues with the drug's metabolism or other biological responses that may not be so evident clinically. The lab has a duty to ensure that they are vigilant for such cases and have a communication pathway to inform those responsible for the clinical study when potentially significant unusual results are seen. Besides conducting research in a safe manner, it is critical that data we generate are fit for purpose. That's to say that they are good enough for our sponsors and regulatory agencies to guide development and make decisions on candidate molecules. But what are good enough data, I hear you ask? Well, it means that our data are sufficiently close to 100% perfect, error-free data to be able to make the decisions that are needed at the particular stage of drug development. Be they go, no-go decisions on the future of the drug, or a decision by regulatory authorities to approve a new therapeutic agent. Note that we are not aiming to produce perfect data in clinical research. The old adage, the best is the enemy of the good, has been mathematically proven and can be applied here. The cost associated with incremental improvements in quality becomes ever higher as perfection is approached and becomes disproportionate to any additional benefit achieved. Importantly, though, we have to be 100% certain that there are no errors in our work that would lead to suboptimal or erroneous decisions being made that could then lead to safety and efficacy problems. That's to say that we can look at quality not as being perfection, but as being the absence of errors that matter, about which more in a subsequent session. More specifically, we have to ensure that our data have sufficient integrity for their purpose. To understand what this means, it is enlightening to go back to the origin of the current good laboratory practice regulations, which, in large part, stem from problems that were uncovered at Searle Pharmaceuticals during their safety testing of several chemicals, including the sweetener aspartame, widely used in Diet Coke and other low-calorie products. This video clip illustrates some of the problems encountered.
The FDA studied not only the aspartame tests of Searle, but 6 other products, and concluded that they were so shoddy and so unsupporting of safety that they actually recommended that a grand jury be convened and that Searle be investigated for criminal violations of the law.
They took liberties with their testing. The work was sloppy in many ways. It wasn't properly supervised, monitored, and the reports that Searle would submit to the government on these tests, they glossed over certain findings which they felt would perhaps put their products in a bad light.
The grand jury pressed no charges against Searle, but the FDA sent in another team of investigators, this time to check 3 key tests on aspartame. Reports from both investigations showed serious problems.
There were animals that developed tumors, let's say, during the course of testing. These tumors were removed by Searle from the animals, and they never informed the FDA that they had any such tumors originally. Here's the sort of problem that we encountered with Searle records. If one follows any given animal, and one can see that the animal died at a certain date, but at a subsequent date, that animal was found to be alive, it died once more, it came alive again, died for a 3rd time, and so on and so forth. Now, we don't really believe in reincarnation, so what must have happened is either that animal, once it died, was replaced by a live animal, which had not been exposed to the chemical on test, or else the technicians or the observers that are supposed to make these observations cannot tell the difference between a live and a dead animal, or else their records are hopelessly fouled up. Now, no matter what possibility is the true one here, one cannot have much faith in the reliability of a study such as this.
You know, some people say
only nerds drink Diet Coke.
So, according to the test results, Diet Coke may be more beneficial to us once dead, although this doesn't make for a snappy commercial, and Coca-Cola have concentrated on more positive lifestyle messages in their campaigns.
Think the things that solve the problems that change the world, that ultimately result in making cha-ching. Those nerds do it smarter and do it with a Diet Coke in hand.
get smart, drink up.
We can, though, easily distill some fundamental compliance principles from the Searle video segment. Data have to be consistent. Results that contradict each other undermine the credibility of the data. Where there is inconsistency, this should be investigated and resolved to ensure the accuracy of the data. The data should be recorded in sufficient detail to allow reconstruction of the experiments, knowledge of who did what, when, how, and the reasons for any changes need to be evident from documents. Data have to be complete. Important findings or observations cannot be glossed over or not mentioned, like the tumours that were removed. Personnel training has to be adequate to ensure the data are trustworthy, that staff can differentiate between dead and alive. A state of control is necessary to avoid records being hopelessly fouled up. In fact, the core concepts of data integrity are accuracy, consistency, and completeness. And when evaluating issues and data, these should be used as yardsticks to conclude on the acceptability of data. So, to summarize this first session reviewing the principles of GxP compliance, we have looked at two ideas that maybe need to be combined so that we can use them in practice. The first idea is that of fitness for purpose, that we must ensure the absence of significant errors. The second segment illustrates what happens when studies are so riddled with errors that matter that their conclusions cannot be evaluated or trusted for decision-making. How do we reconcile these ideas and use them in our everyday work? One key is to apply risk-based thinking. A simple 5-step scheme will be introduced now and reinforced in a subsequent training. These steps being: firstly, describe the issue, what happened. Then, describe what could go wrong as a result. What are the consequences, say, of some missing records? Thirdly, define the severity and impact of these consequences. What effect do they have on the study data and study conclusions? Identify what measures are already in place to prevent this type of risk from occurring. If they failed, then why did they fail? And lastly, identify further measures you think could be necessary to bring the risk to an acceptable level based on the nature and severity of the issue. In thinking about and understanding severity, it helps to look at the types of errors that could matter to regulatory agencies, issues that could delay or lead to study rejection. This EMA guidance document and AAPS journal article describe such potential inspection triggers and errors that matter. A scan of these shows that EMA and FDA are most concerned about inconsistencies in data, indications of lack of control, such as large numbers of reassays, missing data, or data that don't make sense or fit existing models. Whatever role you play in our work to get medicines to patients, issues that arise should be thought about in terms of how they affect adherence to the fundamental compliance principles described here. For example, in the lab, are you excluding any data which had a bearing on the results for the study? Use of a simple risk-based thinking format will help make the right decisions on situations and ensure that we do not encounter errors that matter, that we have not anticipated, avoided, or remediated adequately. More to come on these topics soon.